Hello, I'm Meredith Blackwell, and I'm here at International Mycological Congress 12, and it's in Maastricht, Netherlands. It's the 13th of August, 2024, and I'm going to talk now with the person who has been highly involved in this Congress, but also to find out about not just the Congress, but about his career too, because he's had big jumps in his career. And this is Wieland Meyer. Wieland?
Hello.
Nice to see you.
Nice to see you, Meredith.
So, could you start telling us about your life? Where were you born? What sort of family? Did you get interested in fungi as a small child? What happened?
Well, actually, I was born in East Germany, in Zwickau, which is close to the Czech border. And when I studied, or I wanted to study, originally I wanted to study medicine, but in a socialist country, I was told, as a Christian, you cannot study medicine. So I made a complaint to the government, and they said, you study biology. So, okay, I got studied biology, which I'm probably happier now than if I would be a medical doctor. And so that's how I came to the biology field. So I studied at the Humboldt University in Berlin, biology, first plant physiology. And then we got a very young, dynamic new professor who looked at genetics, and then I said, it's much more interesting genetics than plant physiology.
Who was that?
That was Thomas Bonner from East Germany. And when I finished, we had, usually I was asked then, we didn't have a Bachelor of Science. We had a 5-year diploma study. We were more or less similar to a master degree. And then you had the time to actually make a decision. You go to work, or you do a PhD. So I decided to do a PhD, and
was it easy in that?
It was easy. It was relatively easy. I think we were very few students, but I think in East Germany we had free scholarships, so studies was for free. We had free scholarships, and I think we also had, because there were so few students in this field, because the selection process was done before, we all had also a chance to find a job. So after the 5 years, you got a job, and in my case, I worked for a biodiversity center in East Germany, in Berlin, and they said, well, we really want a new vector construction for yeasts, and maybe you can do this as a PhD, but you also work already for us. So, but they don't have actually a research place, so they moved me back to the university. I went back to the laboratory where I did my normal diploma work, and I started for 2 years to do yeast genetics. And then I thought it was kind of not really the right thing, and there was an opportunity where our head of department was asked from a factory which was in East Germany, kind of a concern, and now it was a huge agglomeration of companies who did beer and wine productions. And they had the idea to use new molecular techniques to type their yeast strains for fermentation for patent reasons.
So what year was that?
That was in 1987.
So just before PCR.
Yes, it was just before PCR, and then I think when I was I started to, and that was around the time, it's a very good question, around the time of PCR, because I went in 1989 when we had the changes in East Germany, just when it started to be kind of politically relevant. I went to the Soviet Union, to the Academy of Science, and there I actually saw the first PCR. Somebody sits with a tube and goes 1 degree, 2 degree, and goes back and made a PCR. So that's what the people actually did. They had no machines, but they had different water passes.
And probably no enzymes from Yellowstone National
Park.
No,
they had an enzyme made several. This time we went to Vilnius, which is now, I think, what is this? The major company who makes enzymes.
I don't know.
Yeah, it's now taken over. There was a company in Estonia and Lithuania which made enzymes, and we went there usually by plane to take a whole tube and come back to the laboratory.
Oh my gosh.
It was not nicely packed as now. It was Fermentas now. Now it's Fermentas, which is the company. And so I changed, and I think that was a very good choice, because I was doing, at the time there was Alec Jeffreys developing fingerprinting for human gene typing, and then I picked the technology up and said, let's do this for microorganisms. So I was probably one of the very early genotyping people for fungi. So first we did like DNA typing, and then I moved to PCR-based fingerprinting, and then from this on I think we developed lots of typing techniques.
Yeah, I really resented it when I'd spent 20 years trying to learn fungi, and then some undergraduate came in and within a week was doing identification of the fungi.
Yes, that's probably how it was. I think I was doing for the first time, I think, typing from different strains, from different people, like maybe people like you who worked on taxonomy of specific groups. And I worked on penicillium with people from Greifswald, and I worked with people from other parts, and so it was not only one field, it was different organisms. That's why maybe also this strange career I had, partially medical, partially not medical, because I was probably one of the few mycologists who worked like in both fields and overlapping on both sides. And then the change came, the wall fell down, and the first congress I went was in Lausanne. And I met John Taylor, actually not John Taylor directly, I met Don Reynolds, and also Ridas Vulgalis.
Okay.
And both of them said, what are you doing? Don't you want to do a postdoc with us? And then I wrote to John Taylor, and I wrote to Tom Mitchell. And Tom Mitchell at Duke was immediately answering, yes, you should come, we would like to do this. He just came back from a sabbatical in Berkeley where he learned sequencing, and he was really keen to get some molecular biologists.
And that's about the time I went to Berkeley and learned PCR from John.
Exactly, so I think probably that's what happened. So and then I went to the US. I think I ended up in Durham because John Taylor was not answering fast enough, so Tom Mitchell was fast. And then I actually started to work with Ridas and Joe Heidman later to look at Cryptococcus. And we did actually, Tom was very much interested in my DNA fingerprinting and in PCR fingerprinting to look at the epidemiology of cryptococcal infections in the US. And we found actually that there's a large genetic diversity on Cryptococcus. At the time was the outbreak of HIV and AIDS, and there was a lot of co-infections with Cryptococcus, and so they were interested in the medical center to look at how we...
And I remember there was some very early reports where people thought there was a fungus that was causing AIDS.
Exactly, so there was this, and it was then, were they all the same, were they clonal or were they variation? And it was actually, our work then showed that there was a large variation, and it also showed then over time, this 3 years I was at Tom's lab, we looked at lots of isolates from the world of Cryptococcus, and we realized it's not only a single species, it's probably more than 2 species, and over time I now my whole life involved that we have now maybe 10 species which were before one, based on this genetic variation. And I developed from the fingerprinting to DNA fingerprinting to multilocus sequence typing and then genomics to look for this.
Okay. I think I first met you in Oslo, walking across the campus, but then I really knew you better when I went to Australia to a wonderful International Mycological Congress there. I really enjoyed that.
Thank you.
Yeah. So you were in charge of that.
Well, yeah. So I think in Oslo actually was to bridge the gap. So from Duke. We had a visitor from Australia, Tanya Sorrell, who came to visit Tom Mitchell's lab, and they wanted to collaborate, but Tom had not so much interest of large collaboration at the time. And then she asked, why? If he doesn't want, let's invite the postdoc. So she invited me to Australia, and then after half a year, she told me, you don't have just a postdoc position, you have a permanent position. It was a total surprise. I came with like one suitcase to Australia.
That's very strange that you didn't know.
Yes, and I stood then like 30 years there. But I think then I became involved also. I was the first non-classical mycologist who became president of the Australasian Mycological Society. And then they asked me in Oslo actually if we could bid for the next congress in Keynes.
Okay.
And so then I was in charge of developing the program and organizing IMC8 in Keynes. Yeah, I think it was one of the major congresses everybody tells me was nice.
It was wonderful for me because I went to the Daintree Forest
Yes.
before, and I saw a taipan. It ran across in front of the Jeep I was riding in, and it was very scary. But there were many mycological things that impressed me, but it was that taipan that got me started before the meeting. There was also a nice art exhibit. I have two paintings I bought there and shipped home.
Mm-hmm.
I tried to get them unframed, and the people said no, by the time you would get them unframed and roll them up and get them framed at home, it would cost as much as the shipping. Yeah, so I sent them with the frames.
So you have some Aboriginal art now. Yeah.
Well, they're not Aboriginal.
No.
They were people. There was an art show from artists there in Keynes.
Mm-hmm.
Yeah, so it was very nice. Yeah. Yeah. So you stayed there 30 years.
I stood in Australia finally 30 years, and I developed a mycology reference laboratory for medical mycology at Sydney University. And in parallel, I think we built up a laboratory which does research. Mainly it did initially population genetics on Cryptococcus, but then we also looked at Scedosporium population genetics. We looked at, by doing this, we also find out that some strains of Cryptococcus were more virulent than others, but it was the same species, even more or less the same genotype. So then we were looking at genome analysis to see what is the difference, why is one strain more virulent than the other. And I think we started also to do lots of animal experiments in mice to see the response of the infection, and we realized also that maybe it has something to do with the host response also. So I did more than just epidemiology, so we went in this direction, and we also looked then at databasing. So in Tom's lab, I was doing two projects. One was phylogeny of yeasts, and this was a little bit in parallel to Clade's Kurtzman's work, which Clade was faster than as Tom. But it spent my whole life that I looked at phylogeny and yeast with my group, and we used this then later for developing techniques for early diagnostics in fungal infections of humans. Because classical diagnostic takes weeks before things sporulate, and usually the patient is then already in a very bad shape, as we have seen today in one of the talks. So we're trying to do molecular identification, and now the latest things we're trying to do is metagenomics, to sequence the whole sample of the human within a few hours, and hopefully can give the clinician a much more guided response of what organisms are present. The challenge is here now that we're missing the databases in the background, that we don't have actually all genomes of all fungi. So now we may be able to do a lot of DNA sequencing, but the identification is stopped because we don't have the reference databases. So that's really important. It came out that we could identify the organisms which are in the sample, but there were also other organisms, because the homology between fungi is quite big, and if you're not in the database, your organism, then it gets the next closest one.
Right.
And then the clinician has maybe not one organism, but 5 to choose from. And then it becomes really important how to do this.
Yeah, it always has bothered me if you get something, you get a hit, but then you don't know that that's the closest relative, and you wonder, was something described, or 5 things described, and they're not, the DNA's not done. So
exactly, and
so
also
we're going to forget all these fungi if they don't have a DNA sequence.
Yeah, that comes back to the discussion we also have here now. What are we doing with DNA sequences? But also we need to be careful, because there's really not enough knowledge there to see what is the genetic diversity within a species and between species. So that was some of the very early questions we had in Cryptococcus, to figure out, is this different species or not? And then when we defined the groups, which were quite clearly different, then the question was, is it good to describe a new species? And what do we do in the clinics if we can't identify them?
And Cryptococcus sometimes is not Cryptococcus. You've got lots of genera involved too. Huh?
Yeah, there are no new genera now, exactly. But also I think it's the way how to identify this. If we have maybe different species, but they all react the same to antifungals, then the question for the clinic is, do I really need to know what organism it is, or do I just need, is it resistant or susceptible?
Are there new therapeutics being discovered or found?
In the recent years, I think there are a number of new chemicals, molecules in test, which hopefully give an alternative, because we unfortunately see resistance for a number of antifungals developing in fungi, so that there is a big need to get alternative molecules to actually combat this. And there's maybe a handful now which seem to be promising, but as you know, this takes quite a long time. They have to go to clinical trials, and before they go to a production. So I think we still have only maybe 5 to 8 antifungals which are available, and that makes it really kind of limited what you can do.
Okay, now this new fungus named for the ear, aureus?
Candida auris?
Yeah, and it's very important because it's antibiotic resistant. And so is it resistant to all antibiotics known today?
No. So I think, so when Candida auris is interesting. I think for a long time, we only know about infections and the hospital, the nosocomial infections, that there are many infections at the same hospital. So there was a reaction quite drastically that they were resistant. So first of all, they're not all multi-resistant. Not all strains are multi-resistant, but yes, quite a number. So it's really a threat. But we also, for years, on some of the new drugs, they are susceptible, luckily. But I think for years there was no environmental niche there. So people always talked about, for maybe the first 10 years, this is a fungus which only occurs in hospitals, and it was like really extreme. People had to take the collar off the walls because they felt it's all spread and things. And I was always very skeptical about this. So recently, I think we did a kind of a study where we tipped into the NCBI database, and I was thinking we should have all these metagenome data now, the meta barcoding data even, from soil samples. And I thought, well, why don't we just screen the whole NCBI meta barcoding database and see if we can find traces of DNA for auris. And we did so, and it crashed NCBI. They were not very happy that we had so many searches, but we found a solution via search bases on the cloud to do this. And not surprisingly, we found actually a number of environmental samples where Candida auris was present. And this was, of course, only theoretical because we have only molecular data. But actually, 2 years ago, an Indian group picked my idea up and said, look, is this in water, is it there and there? And they went out and they could actually for the first time show it's actually in Blackwell near the Andaman Islands in India, and they could identify auris from there. So I think it's actually a nice way of recycling all this huge data which we have to find new ways to at least give hints. Where do you have to look for an environmental niche if you don't have an idea when you start?
Yeah, we're going to talk later this afternoon to Conrad Schock, who's here, who works at the NCBI.
Exactly.
Yeah, so that'll be of interest. I've worked with several fungi that, well, this past year, one of them called Lobosporangium, which is it's an old zygomycete that doesn't produce zygospores. But I found it as a graduate student. And 2 years before, 2 other people had found it. And then in the intervening 40 years, no one's found it. And so I'd offered a prize. And lo and behold, somebody found it in a dry environment. And I think that our data, they didn't know about it, but our data would have helped them pinpoint. And they did find it in a desert-type environment. So it was interesting, you know. And we used to talk about the mother lode. You had to find out where the mother lode occurred.
Yes.
And then you had all the fungus you needed.
I think it's actually interesting how many data we have which are unused, because people do a meta barcoding study in a certain field and they look for certain organisms, and their analysis only focused on the organisms they are looking for. They disregard all the other DNA sequences which are there. So I think maybe if you're much more open about looking at all the reads in these meta barcodes, I think we find actually a lot of new clues for new ways of directions of research.
Yeah. So you were in Australia. Now you're back in Europe.
Yes. So I think in Australia, after I was there like 29 years at Sydney University, where I was the head of the medical mycology group, I worked very fantastic with people in the medical field. was in between the General Secretary of ISHM also, and then became Vice President and also President of IMA. Then the opportunity came, well, there's this fantastic institute, Westerdijk, before Central Bureau for Schimmel Cultures, and I worked with them actually since my PhD. It's a very interesting story actually. I did work on Trichoderma, on some strains of Trichoderma from the University of Vienna during my PhD, and published a paper, and then the wall fell down, and there was this meeting in no, it's not in Seattle, the next in Oregon.
There was an MSA meeting.
Newport,
Yeah,
where
was
the
there.
Holomorph Congress was organized by Don Reynolds,
and John Taylor.
Yes. And I met somebody very famous, Walter Gams.
Yeah.
And Walter came to me and said, "You are the author of this paper. This is all wrong." And I said, okay, sorry. I got 10 tubes of DNA. I have never had the fungus seen. And you better contact the people in Vienna to give you the strains, and then we will look at this. And that started a long collaboration with Walter. And I went to two people. I went to Gary Samuels at Beltsfield, and he had prepared all the Trichoderma strains, and then he had all these figures there, and he moved them around and said, okay, I was like, okay, I'm a molecular biologist. If you as a mycologist have problem to figure out what species is what here, how do you really do this? And then Walter invited me, and Walter set me on a microscope. He had prepared all these Trichoderma strains, put me on a microscope on a drawing tube, and then I was drawing these fungi, and they looked all the same. And he said, but you need not to draw what you see. You need to put your knowledge in the difference. I said, okay, sorry. So but then actually we worked out there was an exchange of two tubes. And when we find this out, they said, it's all fine. Your study's all fine. It's just two names have to be changed. But it actually, what is really important for me was to learn from this. It's really important to look at the fungi and not just on the DNA. And from then on, I asked all my students to actually also make pictures of their fungi. They at least know what they are working with and see this. So I think it's very important that we have not only molecular data, but we also have morphological, physiological data, environmental data before we make decisions on new species. So that's, I think, I learned from this very early kind of experience.
I wrote a textbook, helped write a textbook at the time all of this was happening. And there's one huge mistake in it, and it was because we used a paper that someone had mixed up tubes. And so we've got the wrong story for one kind of important fungus. So,
exactly, this happens all the time, and I think it's good to start from the beginning where you do your isolation, that you know what you do.
Yeah.
But coming back then, so after the Australia, I thought, wow, this opportunity came up, and so I thought, wow, I'm now in this level of my career where I think I can do something more to the mycology community. And when the director position came up for Westerdijk, I thought that was my motivation, that this is a fantastic collection. I can use this to develop databases which we need for reference. And if this institute is not doing it, who else does it? Because we have the correctly identified strains. So that was one of the motivations. I say we can do DNA databases, we can have genome databases, we can develop multi-bank and multi-DOF databases for proteins, because we have the isolates and we have the technology. So that's one of the things which attracted me to the Westerdijk. So now since 1 year I'm in the Westerdijk, and I think we're trying to reshape the institute in a way that we can accommodate these modern taxonomy requirements. And I think maybe taxonomy in a way should be more like a holistic approach from all sides now, based on genomics and then comparison to the physiological data and the morphology to figure out, is there the boundaries and what is really different. Maybe in the future, if we look at, we have 12 billion fungi and we have 150,000 described, maybe some AI technology with the right database behind can help then the human to accelerate this, and then with the human brain correcting AI mistakes, we're making maybe better progress than if we do one by one description.
I'm glad I caught that higher 12 million.
Yes, exactly.
That number. Some people use lower numbers, but I like the higher numbers.
Yes, exactly.
Anyway, it's a new world, and I'm glad I was here before PCR. And because I think I can appreciate more what's what's happened in the last 20 years.
Yeah, but it is also for me as a director of this institute, I think I would like to have also the taxonomy maintained.
Yes.
I really feel like it's really important, and I think we need to have both sides of the coin.
Definitely.
To have actually really good taxonomists, and we have then the molecular biologists who supplement each other to do this.
Well, I think when you have a phylogeny, that helps direct what you're doing with the morphology. And I like electron microscopy, and
Yeah.
so many people have stopped using TEM.
Yeah.
But so what do you think about using DNA for touch?
When I look at, I listen to the two discussion points, I'm actually a little bit concerned. So I'm a molecular biologist, I'm all for molecular biology, but it depends what locus is. So if the first proposal which says we can use any DNA locus, any kind of DNA sequence, I'm skeptical, because you need actually a locus which is discriminatory, which you have a database and which standardized, that people are not sequencing one part and comparing it with something else. So that's the first proposal. I'm not really supportive for this. The second one, I'm a little bit misguided, because it says genomic DNA. I feel like if we have the whole genome, that may be something good, but if we have genomic DNA and we still have big chunks, but different chunks to compare, you may have two different species described from the same species because they don't match. So I think we need to be very carefully where we go, and I think in the long run we now have the technology to have a single cell genome done. So maybe it is sufficient to do. It will come cheaper. It's now very expensive, but it will come cheaper. So I think if we're in this situation, yes, I think that probably would be very helpful. But any kind of sequence, I feel it needs to be more standardized on loci, which the community agrees to, and also which has a database behind it, because you need to. On the other side, I feel like we need to have databases which maintains all these undescribed DNA, because if we found in one study today a piece of DNA which we can't, for example, I'm now involved in some studies from Antarctica where we found a number of strains which have no homology to whatever is described, but maybe to a genus, maybe you can do this. So if we just place it and we don't have a description, we need to maintain this sequence, because somebody else could find exactly the same somewhere else. And so we need to find a way how we can label these DNA sequences, that they are available for the community, but maybe not already totally described as a species, but when we have the strains, that we can link them up. I think that's a challenge, because we generate so much sequences. It would be a pity if we describe 3 or 4 species from the same material just because we don't have the databases to link them up. That's one thing.
But we used to do that with morphology.
Exactly.
Anyway, eventually we hope to link it all up.
Yeah.
But I must say that CBS, which I started knowing it as CBS, that this has been a wonderful resource for the entire world. And in the United States, we had a Tree of Life project, which was funded by our National Science Foundation. And where did we go for specimens? Here. Not to any resource in the United States, because there's a totally different business model.
Yeah.
And so it's so much easier, and there's so many more fungi here of all sorts, and they're well curated. So.
Yes, I think that's actually the challenge I have also with the Academy of Science, who is the host organization of our institute, to tell them, look, there's a lot of funding needed. It's important that you have this collection is the gift from the Netherlands to the world, and it's kind of a national heritage. It's the same level as maybe the Rijksmuseum or the Van Gogh Museum. The funding should come to this, because this is a resource which is so rich. And also for me as a challenge as a director is to generate funding, that we have the taxonomic fields that we can actually support the well-curated collection in the long run. This is the positive thing we have, that we have very well-curated strains, very well-described strains, but we need to maintain this level. to continue.
And you do that by hiring mycologists who can study them and collect.
Yes, I think I'm trying to hire, to maintain. We have lots of students coming through our institute, and some of them are fantastic new taxonomists. Yeah? And I'm really trying to find ways how we can maintain them, give them a career opportunity. And at the moment, we are on a level where we are going out to the academy to get funding for this, to expand our basic budget to actually enable this young generation to be there and not move on to somewhere else. Yeah, that's my hope.
Well, I think it's a wonderful place to be. And
Oh yeah, that's for sure. It's a very exciting environment, lots of very enthusiastic people, and I think it's also very diverse. So we have now people from taxonomy, but we also have product discovery, we have fungal physiology, we have a lot of molecular work now doing, we have food science, food security, indoor fungi. So it's a fantastic place. If you come there, you find one expert for anything somewhere in the building. So that's what I think is the positive thing. And also I feel like the combination of the connection and research, it's unique, because if you have the ATCC, for example, it's a collection. There is no research attached, you know. So I think this is what many collections are there, but we have this unique combination to have research on the strains.
and they said very trustable. We
Yes,
trust.
yes,
we
thank
trust
you, yeah.
Well, thank you very much.
Well, it's a
I've
pleasure.
enjoyed talking to you, and congratulations on your Congress.
Thank you very much. I hope it's really good for everybody to be here. The feedback is kind of overwhelming. They like it very much, the variation of the topics and the people. I'm very
yes.
glad that there's so many people who could make
Yeah.
it. Thank you very much.
Thank you.